Good day, students. We will look at 3 examples of interactions and interdependency: organic matter decomposition, symbiotic nitrogen fixation, and mycorrhizal fungi. The process of degrading the fresh organic material added to the soil is a complex process. It involves intricate interplay from numerous types of species. If some of these organisms are absent, the decomposition and the related nutrient cycling will be much slower and may stop altogether. Decomposition of complex organic material like plant litter begins with mixing and shredding. Earthworms and all other soil arthropods are very adapted at this. Earthworms pull litter into their burrows and mix it with soil. Then insects and other microarthropods feed on the litter, pulling it apart into small pieces. Mixing the material into the soil brings it into contact with other soil degraders and greatly increase the surface area exposed to degraders. That's very important, increasing the surface area for the decomposition by other interplayers. When fresh organic material is mixed into the soil, bacteria respond almost immediately. They begin to feed on the simple organic compounds such as sugars, proteins, amino acids preferentially. Bacterial numbers then increase very rapidly in response to the food source. But bacteria have a harder time with some of the more complex organic compounds in the litter. And these complex compounds sometimes prevent the bacteria from getting the remaining material they could degrade. It's as if the food is locked in a cupboard, okay? So they start degrading simple compounds first, and the harder compounds for them to degrade are left at the end. Okay, this is also called preferential degradation. So let's bring on the fungi. Another players come into play. Their populations start to increase also more slowly than the bacteria, but they are able to degrade the complex compound the bacteria could not get at. Things like cellulose and lignin. The degrading work of the fungi helps to open up the locked cupboard and give other microbes access to the remaining simple compounds. The final degraders are the actinomycetes. They are the cleanup crew, and they come at the final stages of the decomposition. Like fungi, they're able to degrade complex compounds like cellulose, lignin, and chitin. No, not to be forgotten are the protists and nematodes. These are the predators hunting around in the soil for the creatures that got fat from eating the plant litter. They feed on the bacteria and on the fungi that their population already started to grow, and they start releasing nutrients into the soil. So this is also beneficial in the nutrient cycling. Degradation of organic material involves an important balance between two important elements, carbon and nitrogen, in the material being degraded, in the degraders and in the soil. When fresh litter is degraded, okay, about 2/3 of carbon is released as carbon dioxide through respiration, and about 1/3 goes into building new biomass. This cycle repeats over and over until the material is degraded to a stable soil humus. Okay, so the cycle goes on and on until the degradation process reaches a stable soil carbon. Bacteria and fungi, they have an average carbon to nitrogen ratio in their cells about 8:1. This ratio must be maintained. If fresh organic material has a CN ratio of around 24:1, this provides exactly the ratio needed to keep the bacteria and fungi C to N ratio at 8:1. But This is because 2/3 of the carbon being lost as carbon dioxide, 2/3 of it is lost as CO2, and so the remaining, the CN ratio of what the microbes actually use is very close to 8:1. So the microbial ratio is maintained with no uptake or release of nitrogen. Okay, and here if the 24 added liter is at 24:1, 8:1 is maintained on carbon biomass, and 2/3 is released as CO2. Okay, if the liter changes, if we add liter, so let's say it's around 90:1, the microbes will be taking up material with a CN ratio of 30:1. Okay, this is much too high in carbon. They need nitrogen and will scavenge nitrogen from the soil in a process called nitrogen immobilization. Because microbes are much better at grabbing nitrogen than plants are, plants become nitrogen deficient. In general, if the litter's carbon to nitrogen ratio is above 30, there will be a net immobilization will result. Okay, so they will start to take the nitrogen from the soil, and there will be an immobilization. If the litter has low carbon to nitrogen ratio, say 9:1, the microorganisms will be taking up material with a C to N ratio near 3:1. This is much more nitrogen that they need, and the excess nitrogen will be released into the soil as inorganic nitrogen in a process called mineralization. Mineralization will usually occur if litter C to N ratio is less than about 20:1. Okay, so there will be a net mineralization if that happens. So soil will acquire nitrogen from the microbial degradation. Related to nitrogen availability is the symbiotic nitrogen fixation, and the nitrogen fixing microorganisms, a well-known process to farmers worldwide. Farmers, they like the idea of nitrogen fixation. Many bacteria, they have the ability to convert nitrogen in the atmosphere into inorganic nitrogen that plants can utilize. This process, however, can be made much more efficient if the bacteria don't have to go searching for food to keep themselves functioning. Therefore, some species of bacteria, notably the mycorrhizae, the rhizobia, they have developed symbiotic relationship with roots of leguminous plants. The plant roots keep the bacteria well supplied with sugars that they need to thrive. Okay? In return, the bacteria busily get to fix nitrogen and supply it to the plant. So it's a benefit for both organisms. Another symbiotic relationship in the soil, that of the mycorrhizal fungi. It is maybe less familiar to farmers. Although many fungi live by degrading organic material in the soil, there are also several fungal species that rely on a close association with plants for their livelihood. These are known as the mycorrhizal fungi. The term mycorrhizae means fungus root. These are symbiotic relationship because they benefit both plants and fungus. Some of these fungi live on the external surface of roots, while some actually invade the root cells of the plants. Shown here is an example of the fungus that has invaded the cells. Here we could see it in the green structures. This is, let's say, the vesicle. Okay, and also we could see clearly the arbuscules forming here. So this is the avascular mycorrhizal fungus. They form arbuscules inside the root cells where there is an exchange of nutrients provided by the fungus and sugars provided by the plants. The vesicles, they are storage organs formed by the fungus. These types of fungal root associations are formed almost all important agronomic crops. This is very beneficial symbiosis. The fungi benefit from the association with plant roots because they can feed on sugars produced by the plants. Because of this, fungus does not have to compete with other soil organisms for its food. The plants benefit because in return they receive nutrients and water from the fungi. The fungal hyphae are able to reach much greater volume of soil and the plant root can. In many cases, they extend 5 to 10 centimeters beyond the reach of the roots. And the hyphae also can squeeze into soil pore spaces that are too small for the root hairs to penetrate. In many cases, the fungi are better at extracting nutrients from the soil and than the plant root. This is especially true for phosphorus nutrient, which is very limited in the soil, especially in low fertility soil. The fungi also bring water to the plants. Shown here is an example of the beneficial effects of mycorrhizal fungi on the growth of Douglas fir seedlings. Okay, so here you could see with no mycorrhizae in here, there's no mycorrhizae. And on the second hand, with mycorrhizae, you can see the plant height is much larger than the ones without mycorrhizae. And it is true for most plants having to do associations with mycorrhizae. Also, another benefit that mycorrhizae have, that those fungi generally have a strong influence on soil structure. Their hyphal strands help to hold soil aggregates together, and they also excrete organic substances that help cement the aggregates. This is demonstrated in these photos. So you could see the one on the right and the left here. It's the fungi with the mycorrhizae. They help forming the aggregates. Here's, like, for example, this is also aggregate, and these aggregates in water there can form a water stable aggregates. While in return, in a picture on the right here, well, those aggregates without much fungi, they do not maintain a good structure in water. Okay, so their structure is lost and they don't hold on with shaking with water. Several soil environmental factors affect the growth and activity of soil organisms. Some of these are factors that are altered by soil management. In general, as soil organic matter increases, so too does microbial growth and their activity. The type of organic matter will have some effect on the type of microbial community in the soil. The residue of a single crop may be favored by a certain microorganisms. In a monoculture of that crop, the favored microorganisms will be predominating. They will start to increase and start to multiply. Microorganisms, they are sensitive to oxygen status. Most microbes that are beneficial to crop production, they are aerobic, or they require oxygen. Aerobic organisms will have a hard time thriving in a soil that is frequently flooded. To thrive, microorganisms require adequate moisture and are most active at temperatures ranging from about 20 to 25 degrees Celsius or 65 to 100 degrees Fahrenheit. If the soil is too dry, too cold, or too hot, microbial activity will slow considerably. Finally, soil fertility, especially adequate calcium near neutral pH, will favor the growth of most desirable microorganisms. Agricultural and soil management practices can have a significant effects on soil microbial communities. In general, as the above-ground ecosystem complexity decreases, the soil ecosystem diversity will also decrease. Such Thus, soil microbial diversity in a crop monoculture will likely to be much less than that of a forest or a grassland. The numbers of few species may be much larger in a monoculture. Crop rotation helps to increase this diversity. Okay, so in monoculture there's a decrease in the diversity, but we can help raise a little bit the diversity by crop rotation. Tillage represents a major soil disruption and tends to decrease the diversity. Incorporating plant residue will stimulate the activity of these microbes that can most effectively degrade that type of plant litter. As was mentioned a moment ago, adding lime or nutrients into to infertile soil will tend to increase microbial activity and their biomass. Adding other organic materials such as manure or green manures provides another type of food and helps to increase the overall organic matter levels. Thus, microbial activity and biomass will tend to increase from this management practice. The no-tillage practice, the reduced tillage or no-tillage practice, will reduce till system, are less disruptive of the soil and tend to build up organic matter at the soil surface. This tends to increase microbial diversity and their activity. Pesticide application have a mixed effect on the soil microorganisms. The chemical may be toxic to some and thus detrimental. The activity of bacteria that can degrade the chemical will be also increased. So the application have variable effects on these microbial populations. There are several mechanisms by which pesticides are activated and lost from the soil. These include adsorption, getting stuck onto the soil particles so they cannot be taken up by plants, or attacked by microorganisms. Other pesticides could be leached, being moved out from the soil profile by water percolating through the soil. Some they start volatilizing, essentially evaporating into the air, or they could be photo-decomposed, excuse me, being degraded by the ultraviolet light from the sun. Okay. The pesticides are organic molecules. They are also represent a source of food for soil microbes, especially the bacteria, because every pesticide has a different molecular structure. Not all bacteria are able to use them as food. But among the millions of bacteria in the soil, there will be some that can start to chew on the pesticide molecules. Because most pesticides are very complex molecules, it often takes several species of bacteria to degrade the molecule. Normal application of pesticides adds a very small amount of organic carbon and nitrogen to the soil, especially compared to what is already present in the soil and organic matter and crop and animal residues. Thus, pesticides are really not significant overall as food or energy source for soil microbes. In most cases, they are simply degraded along with the more important food molecules that microbes are consuming. Shown here is a highly simplified diagram of how 2,4-D is broken down in the soil. The acetic acid group, vinegar, at the top of the ring, okay, the top is open to easy attack bacteria, quickly chew it off, and they produce some carbon dioxide. Okay, they release carbon dioxide in here and water, and then the aromatic ring is much tougher to break. Okay, so here's their aromatic ring. It's very hard to break this down, but bacteria with right enzymes, they come along and break it open, and they start to chewing it up into also releasing carbon dioxide that make up the ring, releasing harmless CO2, water, and chloride, and the remaining simple 4 carbon molecules will also get chewed up until all that's left is carbon dioxide, water, and chloride. So all this structure will be converted to CO2, H2O, and chlorine, Cl-. Once the herbicide is applied to the soil, there's usually a lag time of 1 to 2 weeks during which very little degradation would occur. During this time period, the bacteria that feed on the herbicide are rapidly multiplying in response to the new food source. When their population gets sufficiently large, their feeding really begins to deplete the herbicide concentration in the soil. The rate of bacterial population buildup depends on things like soil temperature and moisture. If it is warm and moist, the lag time will be short. If it is cold or dry, the lag time will be much longer. For good weed control with soil applied or residual action of herbicides, the herbicide concentration in soil has to stay high enough for long enough to suppress weed germination and their growth. This critical soil concentration does not pertain to foliar applied herbicides such as 2,4-D. For soil applied herbicides, we don't want the lag time to be too short, but we depend on soil bacteria to degrade the herbicide so we don't run into problems with carrying effect on the next crop or with buildup of herbicides in the soil. Thus, we want herbicide concentration to drop below the maximum safe concentration by the next cropping season. Buildup of herbicide in the soil could also lead to undesirable crop uptake or movement of to groundwater or surface water, and this is a cause of pollution, and it is not a desirable thing. It is difficult to quickly summarize the pesticide effect on soil organisms because of the huge number and variety of both soil organisms and pesticides. In addition, factors such as soil type, temperature, moisture, pH, pesticide concentration, and duration of exposure all influence effects on non-target organisms. In general, most herbicides have minimal adverse effects on soil organisms. Some may affect algae, which would make sense because algae, they're similar to plants, they do photosynthesis. Insecticides obviously can affect the non-target soil insects. The fauna is affected by the insecticides, and some are known to be toxic to earthworms, and so this is also another factor. Fungicides and soil fumigants have adverse effect on a broad array of beneficial fungi, nematodes, and other soil fauna, in addition to the target pathogenic fungal nematodes. So they must be used very widely, these groups of pesticides. Earthworms are very important to soil animal. As soil animals, we discussed earlier, the overall most herbicides are harmless to earthworms. The triazine class of herbicides, they appear to have moderate toxicity to earthworms. An indirect effect of herbicide and good weed control is the reduction in population because loss of plant cover and food supply to the earthworms. So there's an indirect way that herbicides could affect the number and diversity of also other soil microbes such as the earthworm. Insecticides, they vary widely in their effect on earthworms. Keep in mind that there are numerous species of earthworms that vary in their sensitivity to various chemicals. In general, the carbamide class of insecticides, they're highly toxic to earthworms. The organophosphate class of insecticides, as a rule, are of relatively low toxicity to earthworms. There are exceptions, however. These include chemicals listed here, such as the phorate, chloropyrifos, and ethophorofos, and the others. Most of the natural insecticides, synthetic pyrethroids and the chitin inhibitors, okay, they are not known to be toxic. Through several they have not been tested. Several of them they're not been tested yet. So more research need to be invested in this area. The pesticides with the greatest toxicity for earthworms are carbamate fungicides and broad-spectrum fumigants. The toxicity of fumigants is made worse by the fact that there is little possibility for worms to avoid contact with the chemical. There are things that can be done to reduce the effect of toxic pesticides, reduce the frequency of application, allow few years for populations to come back up to normal levels before the next application. Annual applications of a toxic chemical for several consecutive years will take its toll. Avoid broadcast applications at times when worms are more actively feeding and tend to be near the soil surface. Banned application greatly reduces the contact with earthworms and so minimize the effect of the overall population. Now we have opened the black box of the soil microbiology at least a little bit. Let's quickly review some of the key things that we saw in that black box. Soil, they are highly complex and diverse ecosystems. The abundance of soil organisms is immense. They are characterized by large numbers of organisms and large biomass. There are many different kinds of organisms, thousands of species of bacteria in a single teaspoon of soil. This wide array of organisms carry out many different functions in the soil. Diversity in a soil ecosystem gives it stability and resilience. It increases its capacity to function under adverse conditions. The relationship among soil organisms, they're very complicated and highly interdependent. Some are commensal, some are parasitic, some symbiotic. Many of these relationships are highly beneficial for agricultural soil. Soil management and broader agricultural production practices can significantly affect the diversity and abundance of life in the soil ecosystem. Okay, that's it about it for today's lecture, and thank you for listening.